we can get going, and a few other people might roll in, but um, they might miss the intro and they can watch that again later. Um, and
Sounds
yeah,
good.
really appreciate your time. Really excited to hear about this crazy novel model of Parkinson's disease. All yours.
So good afternoon, everyone. Thank you for the invitation to the Neuropharmacology Seminar Club today. Today I want to talk about a very simple questions actually, which is, can sleep improve Parkinson's disease research? It sounds quite interesting when you put a question this way, whether an animal can improve Parkinson's disease research, which also tells immediately that that means like this is something new species, and whether this new species can either has been developed or has been validated for any disease model that people can trustly useful by in future. So we would like to talk about SAFE as in Parkinson diseases model, not because this has never been done before. It is because there are few things that we have explored and found that sits perfectly as a translational space between the existing current pretty popular rodent species and the non-human primates or to the clinical research. Right now I miss out my pointer. Oh, there you go. Um, so the question that we are asking is, can we build a large animal Parkinson's model that is biologically relevant to the dopaminergic dysfunction? Number two, that is anatomically large and has a gyroencephalic brain, which is quite similar to human, that has got convoluted gyres and circuits providing a large surface areas of the brain, and whether this is practically safe, manageable, and more repeatable. And finally, whether this model can be used to test a varieties of therapeutics and medical devices that are of human applications. So that is the gap this current study is set out to address. As I said in the beginning, as informally, my journey is quite similar to many of you currently listening here in the talk. I'm a trained pharmacist and then did my master's degree in pharmacology before I get enrolled in neuroscience. So my experience has been from the molecular level, cellular level to actually the system level pharmacology, where I currently work as a research fellow, but mostly working in neurosurgical approaches of developing a large animal model and testing various devices that are of human sized, new molecule that are yet to come to the market, but that required a human sized animal model. So this is where I've been working in the last 8, 9 years. So my research interest is actually sits nicely as in translational neuropharmacology, which is an intersection of like a brain, a behavior, and therapeutics. So as I can say, most of you in enroll in your degree as in pharmacology, and some of you may be in future want to involve yourself into a neuro pharmacology degree or in neuropharmacology research, exploring various research questions that has still need to be explored. So the experience, the degree as you learn is so different. I never realized that I will end up into as a neuropharmacologist when I was doing my pharmacy degree. I thought I would be just working in a pharmacy in a hospital with the patient interacting and medications and kind of things. But the journey is so different. When you keep exploring, when you keep asking questions to yourself, like what next, Oh, these are the drug molecule we're selling here, but like where they come from? and then like, oh, it come from here, but how they interact with the body or how the drug interact with the body. And then you decided, okay, this is overall body. What about looking actually at a certain systems rather than whole body, and then focusing your um, interest into a specific areas such as brain or such as cardiovascular or respiratory. This is how you point your research. But in my case, it's all been through the neuroscience. Um, People often ask why Parkinson's disease, and many of you here are aware of Parkinson's disease. This is the second most common neurodegenerative disorder after Alzheimer's. And this is actually more than just the dopaminergic loss. As you can see in the cartoon here, the Parkinsonian midbrain, the dopaminergic neurons are depleted compared to the healthy substantia nigra. And as the disease is very progressive, um, it carry not just the motor dysfunction, but also non-motor symptoms. The cardinal features includes tremor, akinesia, a postural imbalance, and mostly seen in aged population. But the research these days suggests that this is not just the disease of old age. The research suggests now even more and more younger people have started displaying the Parkinson's disease. And there are quite gold standard drug for the treatment of Parkinson's, or I would have to say like early Parkinson's disease, such as levodopa. But what will happen is that over the period of time, the levodopa start to decrease its efficacy, showing the fluctuation of on and off periods, leading to an adverse involuntary abnormal movements called dyskinesia, which has been a huge pushback for this drug. And there are a lot of other applications and treatment arrived, such as deep brain stimulations and focused ultrasound has been coming up as a novel approaches for the treatment of Parkinson's disease. But for all these new approaches, new technique, and new technology, we need to have a model that can actually capture the disease relevant biology, and that can allow the therapeutic testings. Moving from the basic research, preclinical research to clinical research, basic research are basically carried out in small rodents to understand their normal physiology, to understand the mechanism, and also to provide the proof of concepts before we get into the clinical zone. After investing lots of money, lots of effort, and lots of commitment working in the preclinical, 80 to 90% of treatment or the drug molecule, they fail to provide a clear clinical benefit moving into the clinical practice. And one of the reasons for that failure of 80 to 90% in the translational pathway could be the animal model. One of the reasons could be the animal model, and there is a translational gap. It's because the experiment that is carried out in a small rodents in terms of the small brain, which is a lyssencephalic brain, and then converted that into the gyrencephalic brain has been a big jump. So if we could develop a model that has a human scale interventions, that would be an amazing steps, and that could potentially minimize some of this translational gap that we currently seeing in the research. So the question is, does our model reproduce the anatomy, biology, and the treatment challenges that matters in human? As I talk about the different models, there are different models exist, and different models answer actually different questions. Not one single model is either best that can recapitulate all the phenotype that we see in any disease progression or in terms of efficacy. For example, for rodent, this is a standard model. They are highly usable, they are very cost effective and produce highly reproducible and well characterized. However, on the other side, they have a small brain, and then some of the structure that we're looking for are not very clear. For example, in striatum, this is where Parkinson's disease come into play. The caudate and putamen have not been clearly demarcated. So, going from rodents to non-human primates, they have a large and carencephalic brain, and their anatomy is very, very relevant to human. Yet they comes with high cost, and there are a lot of ethical concerns around working with human primates. And specifically for Parkinson's disease, the toxins that have been used over the past years, which is MPTP, and it's found to be very, very toxic to animal as well as the researcher. And there are other animal models exist, such as pig, which is again, which has a large brain. Uh, Human-sized application can still be useful in this animal model, but this animal comes with a limited behavioral assessment, and the most important challenges is during the handling. The handling of this animal is really tough in the lab. So among these various species, the question is, where does the shape now fits here? So the shape, which has a large brain compared to pig or any rodent that we have talked before, or even non-human primates, which is like 10% of the human brain, which is 130 gram. And the sea brains, as you can see here, is quite similar in terms of morphology and the microscopic structure, having this gyrencephalic structure that have a gyri and sulcus convoluted around, gives more or less similar structure to the humans. And when we talk about this neurodegenerative disorder, which requires a long testing and efficacy is needed, this animal has a long lifespan. So the longitudinal studies is also useful in this animal model. And as the skull shape head is quite similar to human, which is around 5 to 6 millimeter thickness of the skull, that's the human skull thickness is, that's quite similar to the shape skull thickness. So if we are trying to develop a medical device that can be implanted in skull for the testing safety and efficacy, then the shape skull is quite similar. And this is also why this is really important. And the ship has been around human. There's for many, many years and civilizations, right? The ship has been a domesticated animal, very calm in nature, very easy to handle. So with all these features, safe give us at least an upscaling the animal model while minimizing the complexity of primates that we discussed before. There's a less ethical concern compared to monkey, and it is a less costly compared to monkey. Obviously, it's costly to rodents. So, are we the first people who are trying to find out that SIP can be used in Parkinson's disease? In short, that's no. So there has been a couple of attempts in 3 years ago. A couple of researchers have tried to use SIP as a Parkinson's disease model. One is in 1989 by Hammack et al. And after 5 years, there was a second study done by Baskin to establish SIP as a Parkinson's model. To best of my knowledge, these are only two studies that have tried earlier to develop SIP as a Parkinson's disease model. However, both of these models failed to develop SIP as a Parkinson's disease because of the neurotoxins that they have used, which is MPTP, and this animal has a severe bilateral systemic toxicity. Even though they are trying to develop a unilateral model targeting one side, there was an overall bilateral toxicity seen in this animal model, and some of these sheep started to recover spontaneously after the recovery period. And again, MPTP is very toxic toxins, and it poses a very high health risk to investigators and caretakers. So our approach is now to develop a model using another neurotoxins, not MPTP, but 6-hydroxydopamine, which is relatively safer, um, and to develop a model that is controlled, not anything as an spontaneous recovery as the model develops. And we want to verify that with three different evidence, one with behavioral evidence, supported by histological evidence, and as well as the neurochemical validation, providing that the SIP is not just to listen, but also to establish a platform that can be validated and can be used by future researcher. So we needed to prove four important things here. First, we want to build a model to create a unilateral lesion in substantia nigra. Second, we want to validate that this model suggests a Parkinsonian phenotype. And third, we want to confirm that there is a clear neuronal as well as neurochemical loss as we expect to see in Parkinson's disease. And the fourth one is to use this model as an reproducible model to test the safety and efficacy of dopaminergic agonist as well as medical device that would be a potential treatment in Parkinson's disease. And our approach is very straightforward. So we build a city-based targeting in this SIP model. The SIP, when first come to us, we will CT scan their head, which will tell us where the location of injection would be. And after that location is identified, we will inject 6-hydroxydopamine in one hemisphere of the SIP brain and leave the animal for its recovery so that the animal return to the normal behavior. After the animal recovered from the normal behavior, we will verify the listening protocol as we discussed before with the drug, with the pharmacological agents, with histological evidence to show that this is anatomically relevant. And finally, with a neurochemical validation that suggests that there is definitely loss of dopamine that can be estimated. As this has not been done before, the very first question that we ask ourselves is, what happens actually when you put the neurotoxin into the sea brain? Is it similar to what the earlier failure attempt similar to failure attempt, or is it different, or even is there anything that actually the animal respond to this molecule? So what we found is that during the earlier period, there has been mild post-operative deficits, which is similar to what the earlier studies has found out. And the animal started to lose weight. That was quite concerning for us as well. But then unlike the previous study, the recovery phase was so rapid that after a week of listening, the animal started to bounce back to the normal weight and they started to graze as normal. And we were then like, hang on, we thought that we will be seeing something here after the injection of toxins into the substantia nigra. But what happened is actually seems like something has happened to the animal, but then the animal seems to be more normal. It is very difficult to differentiate a lesion animal with the control animal. And this asks us, all right, so I think this is telling us that our animal has been um, recovered from the lesioning pretty well, and there could be a targeted lesion without the prolonged disruption of normal animal behavior, which is a positive thing. But then we will have to now verify what will happen if we administer the dopaminergic drug to this animal. Does this animal shows any change in behavior? So we started with the three different independent line of evidence. First one was pharmacological evidence. In this evidence, we want to see that what would be the effect of drug, such as dopamine agonist, D1, D2, or even a mixed dopaminergic agonist to this animal model. And second one was what will happen when we harvest the brain from this animal will there be any dopaminergic loss? Or if not, we will try to do an HPLC estimation and see actually how much of the dopamine has been lost in this animal model. And with that, we've started with an apomorphine. So to those of you who have not heard about this molecule before, apomorphine is a mixed D1 and D2 receptor agonist. So what will happen is that when apomorphine is administered to this Parkinsonian animal who has been lesioned on one side, these drugs will create an imbalance in the left and right hemisphere, creating the animal to turn around, which is a strong behavioral readout. So if the animal has been lesioned, which we thought was lesion, then apomorphines will start giving us that behavioral readout measure, which is the animal will start to turn around in contralateral to the site of injection. That is important things, okay? So as I said before, in all our animal model, we have injected the toxin and left substantia nigra, which is projected to left striatum. So the left hemisphere dopaminergic receptors are very sensitive. They are looking for more dopaminergic input, but that has not been. So when you administer the exogenous dopaminergic agonist to animal model, so this dopamine would be more bind to this left hemisphere. So it will create an imbalance in the left and right hemisphere. Because of that, left hemisphere drives the right hemisphere, making the animal turn around. And which is a clear readout measure for us, like, okay, if the animal is Parkinsonian, then by injecting any of dopaminergic drug, we should see the turning around. If we don't see turning around, that means we have not created animal model. And with that, apomorphine clearly produced drug-induced behavioral response that so consistently with the tested animal into the unilateral dopaminergic lesion. After that, there is an increase in locomotor activity, and there is a number of contralateral turnings controlled to vehicles. And the first graph here shows the pharmacokinetics of this uh, apomorphine. The onset of action started to see around 15 minutes, peak at 30 minutes, and then the overall effect is seen for half an hour, for an hour. So after the success of apomorphine verifying a lesion verification to our animal model, we ask, is this just with one model of one dopaminergic agonist, or can we try another agonist and see if we can repeat the similar results? So with that question in mind, we started asking, what about the ropenolol, which is another drug that has been currently used in clinic for Parkinson's disease. Ropenolol is a D2 receptor agonist, and without surprise, ropenolol also produces similar contralateral turnings in our SIP, unilateral SIP Parkinson's disease model, as well as it increased the locomotor activity. So with this behavioral validation that both of these dopaminergic drugs are able to induce contralateral turning in our animal model. Then from those behavioral and pharmacological validation, we move into our histological validation. And the histology clearly shows that the lesion side has got less dopaminergic neuron loss compared to the control side, which is on lesion side. So there's a dense dopaminergic neuronal fiber you can see here, whereas in the lesion side, there are so sparse. And the mechanism of this is that after the injection of toxins into the dopaminergic neuron, this 6-hydroxydopamine mimic its structure chemically to catecholamines. So these are quickly uptake by dopaminergic neurons through the dopamine transporters. Once the 6-hydroxydopamine are in get inside the dopaminergic neuron, this is converted into the oxidative reactive species, which will then destroy the neuronal content, the lipids, and the protein. So by damaging this dopaminergic neuronal cell, which will lead to less dopamine depletion into the striatum. So TS immunohistochemistry demonstrated to us that there was a marked loss of dopaminergic signal on the lesion side compared to non-lesion side. And we also get into the third line of evidence, which is the neurochemical validations. And we want to see that how much of dopamine is actually lost. The HPLC measurement confirmed that not just the dopamine, but also a dopamine metabolites such as DOPAX, HPA has been reduced in the lesion side of the hemisphere. So the data suggests that there has been around 80% of TH positive cells lost in our model of our SIP model of Parkinson's disease, as well as there is a 81% loss of dopamine depletion in the striatum of this animal model. So what these three lines of evidence converge is that the behavioral, which is drug-induced turnings, is clearly demonstrating that the unilateral phenotype of the Parkinson's disease. And in anatomical aspects, the TH immunohistochemistry clearly demonstrate that there is a dopaminergic loss. And as I said, there was 80% of dopaminergic loss that is recorded with TH immunohistochemistry, and the HPLC confirms that there is 81% of dopamine depleted. So all these three different evidence point out to one direction, suggesting that this model that was created using 6-hydroxydopamine is validated model for dopaminergic neuron. Now, getting into the translation, does that means like now can we replace all other species that has currently been used? That would be really stupid things to do. So the goal is actually not to replace the existing model. Rodent acts as the standard model for understanding the mechanism, understanding the screening, and understanding the proof of concepts in all preclinical research. As different species will ask a different model, the different model is capable of different things. So non-human primate, again, they will stay as the best model for their highly anatomical relevance to the human brain. So they will stay there as the most important species. For shape, we can use shape as a platform rather than just a model. It can be used as a platforms for large brain interventions, for testing devices that are of human sized, and for delivery of drug that are of human doses, and for the longitudinal studies, which takes a long time to carry out, that comes with a lot of cost. And imagine having included monkey in a study that goes for years and years. It's getting really tough. It's getting really expensive, and also include a lot of ethical values. In that spectrum, SAFE can be used as a translational model and translational platform, and will provide that opportunity to add one another model that the existing models cannot answer. Um, well, I've been talking about a lot of good things about my model, but this is not the best one. It also comes with its own limitations. In this project, we have been using only one species of sheep. Again, there are a lot of heterogeneity around the different species, so which we have not done. So this particular project was carried out with an one single breed, only female species. And in terms of behavioral task, we not ask the fine motor task as rats and other rodents are capable of. And we have not asked any cognitive testings in this particular model, which is not yet established. And the challenges is that unlike rodents and monkey, there is a very, very limited brain atlas available for the sheep. There is no comprehensive sheep brain atlas exist as of now. The only sheep atlas that we started to use when we first carried out in this project was the French atlas, and it was written in French, so difficult to understand the French language, of course. And also the atlas is very limited to the subcortical structure. The atlas doesn't even have so many structures that we are looking for. So, and the last one was, as this is a unilateral lesion, is to create a hemi-Parkinsonism model of Parkinson's disease. In reality, in a clinical practice, Parkinson's disease is a bilateral, so both sides started to get depleted rather than one side. So again, currently the model that we discuss is not capable of study the bilateral model. So there are so many limitations of the current model, but still, this doesn't invalidate the model itself. The model still has some capability to do, and this limitation actually will define the next set of experiments. So it is also an opportunity when you know that there is no brain at last exist. This is a niche area where young people, young neuropharmacologists like yourself can come in and start to work with the shape brain at last and be a pioneer developing a shape brain at last. Cognitive testing, again, there has not been done any cognitive task in this model. This is again a new niche area for someone who want to use this Parkinson's disease model and want to characterize the cognitive task. As we know, 1 in 5 patients suffering from Parkinson's disease has a cognitive disability. That's a learning, that's a memory, and many other things. So what next from here? So from the model, we can now move into the translational platform. So I've been telling this as a translational platform because it is providing a lot of other opportunities for us in future. For example, we can use our SHIP model of Parkinson's disease to try to find out the fine motor assessment as we see in rodents. We can start developing a cognitive protocol in our model. As this animal live longer, we actually can find out whether levodopa can induce a long-term dyskinesia in this Parkinson's disease model or not. And second thing is to characterizing and testing the different therapies, such as deep brain stimulation, focused ultrasound, and novel drug delivery, including gene therapy, can be tested in these animals. And again, as I said before, it also provides an opportunity for developing a C-brain atlas using the MRI and CT scans data that are currently available to characterize and optimize the standard coordinates for future people who would like to do a neuroscience research in this model. So the take-home message from this talk is that we have established a 6-hydroxydopamine sleep model of Parkinson's disease. We are capable of saying the 81% dopaminergic depletion with the behavioral validation that the model exists. And this is also the platform for the translational drug devices and drug delivery research that in future people may be keen in. So the SIP is not the replacement for the rodents, but it is a potential bridge between rodents and human when we try to find out to test the human size applications that can be translatable in future. So these are other areas that people have started to look for the SIP, such as brain drug delivery, testing therapies at human relevant scale. And a lot of researchers in the US and Korea, including Australia and New Zealand, now trying to work the novel approach of focused ultrasound in shape. And also they are developing a lot of practically large animal devices for the development and for characterization of the device. And also the atlas. The SIP atlas is something this model will provide as a platform. So with that, I will stop here. Thank you so much for your attention. And I'm now happy to take up your question.
Thank you so much. That was really interesting. Uh, I could actually start us off. One of the things I was wondering is how transient of a model this is. How far out from the injection have you followed the turning behaviors before they're just completely gone?
Oh, that's a really good question, Sophia. The symptoms does not go as we have seen now with the previous attempt with the MPTP. They found that after the lesion, this animal will display the Parkinsonian symptoms and then they goes away. But with our model, once the injection is once the dopaminergic depletion happens, it will stay there. It's like irreversible neuronal death. Then the animal will display the symptoms, but it's never going to be spontaneous recovery for this animal model. In terms of like from the point of injection, how long will it take for us to verify that actually the animal has been lesioned, if that is your questions? So that's usually 2 weeks because this is a neurosurgical approach that is quite similar to the human. So it takes a bit of time for this animal to recover, which is around week, I would say around a week. And within that week, the first few days is very severe that we can see in this animal model. That is because it is like hammering, all your dopaminergic neurons at once with the toxins. So the whole animal or the systems, I think what we think is that the whole dopamine systems just got into the shock mode, with the toxins start to destroying all the dopaminergic neuron. And during that period, the animal has a very signature sort of like a lateral bias, you can see. As I said, like our injection is always on the left hemisphere, as this is hemi-Parkinson. You can use any side, but our is always it's easy to operate for me in the left hemisphere, so I always prefer left hemisphere as an unilateral injection. So after the injection during the post-op recovery day 1 and day 2, the head tilted to the right side, and that is immediate indication for us that like, all right, I think this animal is getting lesioned. Earlier we had no clue. We thought like, oh my god, what happened to this animal? That now the head is just in the lateral. Like, did you damage? Is there any stroke or anything happens? Which took a time for us to learn. But after a few days, then the animal started to get back to the normal. And when you realize, like as I said, it's a fast recovery, and after a week, the animal can go back to the pasture. And if you could just go there and stand, you would hardly see the difference between the normal animal and this animal. So good, So then the animal don't have to compromise their normal day-to-day life in the palm and the pasture as well. And the only way you can check whether this is Parkinsonian or not is with your pharmacological validation. So as soon as you started injecting this animal with apomorphine or Panadol, then this animal will start to turn around. And when you say that turning around, then like, all right, this animal is now hemi-Parkinsonian, which is so reproducible, and you can use them for all those applications that I discussed before.
I also wonder if there are feedback mechanisms on the unlesioned side that emerge over time as part of that compensatory mechanism that lets them get back to normal life. Like if that unlesioned side is taking up the slack, what does that actually look like? I think that would be so interesting to learn.
Yeah, so that would be really interesting to learn. I mean, um, so what we find with this animal, because this animal stays with us for more than 6 months, more than I think I've got animal now, they are like 2 to 3 years of Parkinsonian still with us, for us to carry a longitudinal studies. The first thing is they never now get back to the normal. It's going to be late. These are the Parkinson animal. They don't have a reversible recovery for these things. In terms of compensatory mechanism, the receptors on the left hemisphere, in the left striatum, are always hypersensitive. It's going to be always hypersensitive. And whereas in the right side, which is intact, has got those amount of dopaminergic receptors. So those endogenous dopamine that's been released in the brain will create an imbalance, but not strong imbalance enough for them to, spontaneously showing that turning around. So it has to be with the dose, certain dose, and it has to be exogenously administered enough to see the turning around. The working with another limitation, probably it falls under the limitation category, is that working with this large animal model is that imagine like for a 80 kilogram sheep to turn around is not an easy task, right? Unlike the rodents. A lot of hemi-Parkinsonian rat model, you inject and turn and leave the rat around, and they will turn around like a top in a row. They will have more than 200, 300 turnings per second, sorry, per minute, which is a lot. But in our animal model, we don't see that much of turnings. You can clearly see the animal being turned around, which means you can imagine for a molecule to induce that turning behavior, because the behavior is a result, it's a cascade of event that's happening in cellular and molecular level, which is unable for us to see, and that shows as a behavioral approach, right? So it takes really a great amount of drug required for them to turn around.
I also would like to share a little note from Melissa because she's
Mhm.
not able to talk right now. But
Oh,
she just said, "Thank you for your presentation. It's so interesting that you point out that your sheep model is not a replacement for rodents, but rather a bridge between the gap of rodent to human. Based on your explanation of the three method approach you implemented to validate your sheep model, it seems like a lot of work goes into the process of establishing animal models. Is this true across all animal species used in research, or does the level of validation depend on the complexity of the animal?"
thank you, Melissa, for your understanding that like not a single model is good for everything. So you choose model based on what you try to achieve. And in this case, for us, is because our lab is working with Parkinson's disease, and our lab is also working with medical device. We are the only lab in New Zealand who are working with a focused ultrasound. So we are developing a focused ultrasound to deliver the targeted delivery of drug into the brain. And now we are using it to our SIP as a platform to check the safety and efficacy of these devices. Imagine having that big size of the devices to be implanted in a skull, which is the size that is going to be in future be applied in human. If we don't have this large animal model that has a similar CP skull, thickness, boldness, it would probably be difficult for to test that particular devices. So from here, I don't think I've included that in my talk before. That was the first hemi-Parkinsonianship model that has been successfully developed. And moving from that hemi-Parkinsonian, now our lab and myself, I'm moving into creating a bilateral sheep model of Parkinson's disease, which I left it as a limitation before. Now, my lab and my PhD student in my lab, we are trying to focus on developing a bilateral Parkinsonian sheep model because that mimics actually the true clinical presentation of Parkinson's disease, and we want to see how this will go. So that is the one work that is currently working in my lab. And second is we're also creating a cognitive protocol in Parkinson in our Parkinsonianship, which is a niche area, and this has never been done before. So in another few years, we probably will be developing a bilateral Parkinsonianship model for the first time, and also we will develop a cognitive protocol in our C Parkinson's model. So we're thinking of teammates as of now, but we will see how easy it will be to train our sheep to get into the teammates sort of things. So these are future things to look for.
One very interesting cognitive test that I've heard of that might be relevant to this was looking at manganese poisoning, where wolves were caught at varying distances from a manganese mine. And they had a test where they had to run down a corridor and then take a sharp right turn. And normally the quolls could super easily just take the turn. But when they had manganese poisoning, they would have to slow down or they would collide with the wall when they were going to take their turn. So they no longer had uh, the coordination to
Hmm.
land it, essentially. And I don't know what the sheep version of that is, but I think it would be so interesting to see something about gait or speed. That would be very cool.
Yeah, I mean, this is such a great suggestion, Sophia, because as we creating a model that has a motor imbalance and that has a rigidity and that has a loss of dopaminergic neuron, which contribute to overall motor coordination and performance. Any model that you suggest similar to you would be very useful. And our model, and we call it as a platform, our platform is probably the best things to compare with the control animal. You have a control animal, and then you have got your Parkinson shape, and then you offer both these animal model a same task as you said, and technically the hypothesis would be that the Parkinsonian animals would be less coordinated in order to get into that one. But always the confounding thing is that when you develop a model, this is a model development, it's very hard to tell like whether the model has a specifically motoric imbalance or a cognitive imbalance. We don't know yet. We still don't know yet. Another things that we are also planning to is that what happens if we actually go more than 80% depletion. The reason we started go low, go slow is because we have seen that in the past people have tried to work with a ship model and they have failed, and we just don't want to go in the same pathway, right? We don't want to go. So this is why after those two tests there has never been anyone who decided to work with the ship again until we arrive and like, okay, we will take it from here. We want to take that challenge. We want to create. We try to go we learn from those two studied only available study, and then we will try to safely develop a model. So our 80% model itself giving us a clear pictures. That one depletion and having those three different line of evidence, separately give us a strong validations for the model. From 80%, if we depleted 90%, there may be a different sort of model which we've not gone through, and we just don't want to take that risk, as well, because if the model has not worked, we probably would have increased the toxin and then get into that one. But if it is going and it's so reproducible, and now we have got like more than 50, say, Parkinson in shape, so reproducible, and being available to test these new drugs and molecules. So we're quite happy with that one.
Very interesting. Cody and Ingrid, do you guys have any questions? I don't want to just dominate with mine.
No, thank you though. That presentation was fantastic. Oh, sorry, Cody.
No, you're good.
Thank you, Ingrid, and thank you, Cody, for your attention. I mean, you feel free to ask anytime you decided to know more about these things, or if you are you all in your final year of your PhD?
I
Right. Okay. Anyway, so
Um,
I mean,
so I can answer that question because I think Cody just cut out. So Ingrid's a postdoc. She's in, I think, her second or third year now of being a
oh,
postdoctoral
lovely.
fellow.
My third
And
year.
your third year. And then Cody and I are both PhD students. I'm going into my fourth year. Hopefully,
Right.
there will just be five. And Melissa works as a research technician in an Alzheimer's disease lab.
Oh, beautiful. And for your information, Malaysia, there is another researcher in Australia who has developed a SIP model of Alzheimer's disease. I was expecting you guys will ask like, is because SIP is so widely found in New Zealand and Australia, is that the reason why you work with a SIP? Because I often being asked this question, In New Zealand, SIP outnumber the human population. So there is like five SIP per person. Imagine. So the
That's
SIP actually,
crazy.
yeah, the SIP actually outnumber the human population here in New Zealand. again, which is good in a way that it's very cheap for us to use, and there's less of ethical concern. Because in New Zealand, we can't work with monkey. It's illegal. This is also the disadvantage for a researcher like ourselves. It's illegal to work with monkey here in New Zealand. So we have a collaborator in Spain. So we have actually done a similar studies in monkey over Spain, following the similar protocol.
And was that with rhesus?
And that's with macaque.
Okay. Okay. Yeah.
Yeah, that's one macaque. So yeah, we've been always exploring these opportunities to expand the model and see the platform being used by many people. So if you ever want to work with shape, or if you ever want to work with, yeah, I feel like working with a shape, our lab would have opportunities coming in, every now and then for postdoc or research fellow positions. Please keep watching the space.
Thank you so much. This is so different from what we do in our department, but also so relevant. And so a really exciting talk to be able to have. And just really appreciate your time, especially when you've got a cold and aren't feeling good and it's winter. That was a
Yep.
really great talk, and we really appreciate you. So thank you.
All right. So once more, thank you, every one of you, for your attention and providing this platform for me to share all of my work that we've done here in New Zealand.
Thank you so much.
Thank you. Bye, everyone.
Bye.